Dr. Johnson is an associate professor of statistics at Virginia Tech, and she was previously a faculty member at the University of South Florida and held research positions at U Chicago, UC Santa Barbara, and University of Cambridge. She obtained her PhD at UC Santa Cruz in physics, applied math, and statistics. Her research interests are in statistical and mathematical biology, ecology, and epidemiology. In particular, she's interested in how individual behavior and local interactions between individuals in a population influence population-level patterns and statistical methodologies for inference for a mechanistic model of biology systems. And it's worth mentioning that she was also the recipient of the NSF Award. So now let's welcome our speaker.
Thanks very much, Shao. I'm glad to be here. So I've switched my title just slightly. So I was going to talk kind of more broadly about ecological applications, but I decided to focus it a bit more on vector-borne diseases today. And you'll notice that I'm kind of like you can tell a little bit from my background, I do only have one PhD. It's just a little complicated, okay? I don't have three. It was in the Applied Math and Statistics department, was a new department when I was there, and they were not allowed to award degrees. So I had to qualify in physics. So I did all my coursework in physics, and then I did all of the Applied Math and Stats coursework in as well. And then my PhD was on cholera, right? So it was a bit of a wild ride there for me. But because of my background, I really view myself as a kind of the far end of the applied spectrum of statistics. So for those of you who are graduate students, maybe you haven't figured out kind of where you fit in yet, right? There's a whole spectrum, a lot of different flavors of statisticians, from like the very pure theoretical folks to the very applied. I'm really on that applied end of things. And I really also view part of what the things that I do is a kind of translational statistics. So I help, I feel like part of what I'm good at is helping to translate all of the new methodologies and sometimes existing methodologies so that folks in the applied area understand what they are and they are less afraid of them and they're more eager to be able to use them. So a lot of the work that I'm going to be showing you today is part of different efforts and different collaborations that I've had that have been trying to kind of introduce some of these methods, some of them really basic stuff, so that my collaborators in biology and epidemiology can use them more effectively. So as a result, one of the most important things that I always like to do very first is like to thank some of my collaborators. So this is just a subset of the folks that I worked with on the stuff I'm going to be talking about today. So it's a really collaborative endeavor, and I think this really makes what, this is the part that I really like, is finding people who I can have fun and work with. So what I want to talk to you guys today about are vector-borne diseases. So for those of you who are not familiar with this term, vector-borne diseases are those that are not spread directly from person to person or organism to organism, but instead the pathogen that makes you ill must be spread by something else. And often those are spread by various types of arthropods or insects. So these are some that are here in the Americas and that are spreading in the Americas. And the ones that I'm going to focus on today are these ones here, which are all ones that are spread by mosquitoes. So we also have some that have been emerging, like Zika and chikungunya. And altogether, these create and cause a huge amount of mortality and morbidity, that is decrease in quality of life, et cetera, across the world, including here in the Americas. And if we think about not only the fact that we see them here, but where we see them, if we look at where people catch these diseases, for example, here's a map of where people are catching dengue. So every place that's in yellow is where we've found that there's transmission of a particular virus called dengue. There's a couple of different varieties. This causes a lot of illness. It's spreading all over the place. And what we notice is that the places that it tends to spread are places where it's warm, right? So I have marked on here these isoclines that demonstrate kind of where the average winter temperature stays above 10 degrees centigrade. We're starting to, we're right about here, right, today. This is the first time I've been below this line and then given a talk. But what we kind of think about is that then this might tell us that the environmental conditions in a particular location might be important for when and where we see these diseases be transmitted. And we can think about why that might be the case. So here's a diagram of kind of the most common vector-borne disease, which if we think about is usually considered kind of the canonical one, right? So biology also has kind of canonical cases of things. And usually malaria is the thing that people think about when they're trying to understand vector-borne diseases. So here's the general idea. So let's imagine that we have a mosquito up here at the top, and this mosquito has malaria pathogen. It's going to bite a human, or if it's avian malaria, a bird, that pathogen gets transmitted to that host. It develops within the host until there's enough of it inside the host that another mosquito can come along, take a bite, pick up that pathogen itself. Then there has to be some part of kind of development of that pathogen in the mosquito as well. And then once that mosquito is infectious, we can complete the vicious cycle and we can transmit that disease on again to another individual. And the thing that we note is that everything that happens out here in this mosquito is sensitive to environmental conditions. So mosquitoes, as you guys probably notice, less than where I live, they don't like every temperature. So there are no mosquitoes right now in Virginia because it is really, really cold. So they all die. And then if it gets too hot, which you guys probably get here in certain parts of the summer, you probably notice that there are also fewer mosquitoes when it gets too hot. So there's going to be these temperatures that constrain when and where we see this type of transmission. So the big picture question that the folks that I work with that I'm interested is to try to answer this. How can we predict when and where these vector-borne diseases will be transmitted, when that burden will be high, both now and as climate changes? So I think about kind of these related things like, can we use just temperature? What kinds of data do we need to be able to make good predictions? What times and spatial scales can we make good predictions with different types of data and different types of models? And from kind of a modeling perspective, I like to think about how we can combine mechanistic models, that is something about what we understand about the biology, with a tactical model. Now, y'all might not be familiar with this terminology, so I will explain what I mean. So I think about models as being on the spectrum. And on that left-hand side, this is where we work usually as statisticians. Right, so these are the types of models where the typical goal is to describe the relationships and the patterns in your data. You don't necessarily care why those patterns are there. Maybe if you're doing causal inference, maybe you do, but often a lot of what we're doing doesn't really matter why those patterns are there. On the other side are what I think of as these strategic and mechanistic models, which are explicitly trying to incorporate that understanding of whatever is going on. These are sometimes called physics models, for example. And these are usually things like ordinary differential equations, agent-based models, and that sort of thing. And so we're not usually making predictions, for example, out of that, right? We're often just trying to understand. And so the types of predictions that we make from those models are different too. So I tend to think of it as like in terms of if we were making a model for a forecast, that we would be thinking about kind of short-term forecasts. So we don't have to do very much extrapolation, right, from our strategic ones versus kind of long-term ones, like scenarios. So what if this thing happens, what types of patterns might we see? And it turns out that we also then actually need different amounts of data for these different types of models. So my question is always this: how much data do you think you need for these different types of models? Anybody give me a guess? What is your instinct on what you need? I heard something up here.
A lot.
Probably. So I was going to say that at the extremes, usually it's some. Like if you think about your really simple regression model, like you don't necessarily need a lot of data to fit a line. For some of the strategic models, they're literally building data-free models, which is fine too. For understanding, that's sometimes what you want to do. I think if we start trying to want to kind of get in between, where we like maybe think about these intermediate time scales, and we want to include some of both the tactical information and be able to make good predictions and include something that we know about the biology is that unfortunately, this is where you need a lot of data. And from my experience, it's almost always more than what you have, which as a statistician, you always hate to have somebody come to you and say, can I learn this from my data? And you say, I'm sorry, you can't. And you try to do something else instead. So why do we need so much data in that intermediate type of thing? And it's because we need to do two sets of things. Effectively, we're trying to fit two types of models, right? We're trying to say something about some process that underlies the patterns that we see. And then we have to try to match it up with the actual pattern that we see. So we need to be able to do both the why and the what at the same time. And often the type of data that you have just isn't the right type. So it's actually can be really difficult to find the right type of data and to be able to mesh these two approaches. And then finally, sometimes the missing piece is actually not about the data at all. It's really about the model, because not all models are good for all purposes. Right? We're always building different types of models for different purposes. And being able to figure out how we can use components of a model that was not designed for the thing that we want to do to be able to understand something else is something that could be really challenging, but fun. So what I want to do today is take us through kind of a suite of a couple of different types of models that allow us to learn different things about how environment or like understand patterns in vector-borne disease transmission. So I'm going to start with the side of things that we are probably the most familiar with here, with the tactical side, and then I'm going to move to the mechanistic and talk about how we're trying to think about kind of bridging that gap in between. So to start with, I'm going to talk about something called the dengue forecasting challenge. And this was something that the CDC held a few years ago, many years ago now, actually, when I think about this, about how we can predict the timing and severity of dengue outbreaks. And so what we're trying to do is model this response, which is simply just a time series of reported cases of dengue in 2 locations. I'm going to be just talking about San Juan, Puerto Rico today. And then they gave us a whole bunch of possible predictors like precipitation, temperature, something called NDVI, which is a greenness indicator. It tells you how much foliage and plant matter there is. Things like population size, because it turns out you don't get any dengue if there aren't any people, right? And that sort of thing. And the prediction targets are threefold. So there are 3 things that we want to try to predict. This week of peak incidence level, and total cases. So let's see kind of what this looks like. So we have these 2 sets of time series that we're interested in. The top, which is the Puerto Rico, and the second one is from Iquitos, Peru. So those are our 2 locations. And if we kind of imagine lining up every season, so if I'm going to cut this apart by year, I can like line them up from week 0 to week 52. And I'm going to be trying to model 3 different types of things. So the first thing we're going to want to do is to figure out the peak incidence, and that's how high the highest observed case is in any given season. And then the next is the peak week, so the week at which that peak incidence occurs. And then the total cases, which is like our integral underneath over that curve. It's all the things that we saw. And when we were doing this for this competition, we took kind of an ensemble approach, and we did 2 types of models. I'm not going to talk about the first one today, although it's one that all of you graduate students could probably implement yourselves. It's just a simple GLM with like a really simple autoregressive component, sine, a cosine, and some predictors. Making forecasts out of that is a pain in the butt, but it's possible. So instead today, I'm going to talk about this one, this component of it, which is a Gaussian process model. And this one is kind of nice because actually we're not going to use any of those predictors. We're going to use the dengue incidence, some things I'm going to talk about, predictors that are casually observed relationships. You'll understand when I get there. And it's nice because it's fully analytic, and then we add some kind of heteroscedastic extras on top of that. So that's kind of an overview of this first part. So for those of you that are not familiar with it, and I'm often trying to explain these to biologists, so I try to make it as simple as possible. So Gaussian process is a very simple extension of our standard kind of normal regression model, where instead of saying something about what we think the parametric mean is going to be, we're instead going to allow that to vary and be kind of discovered as a special average over kind of nearby points, that your prediction for any location depends on the response nearby in whatever your predictor space is. And in this case, what we wanted to do is we went ahead and we were modeling the square root of dengue incidence. So we have a few zeros and a lot of noise in this data. And our first instinct was that we wanted to take care of the heteroscedasticity that way, like it's your standard transformation. And then we have this Gaussian process. And our Gaussian process, the sigma, the correlation depends on how close the data are to each other in some covariate space. And I'm going to tell you in a moment what our covariates are. For those of you who are familiar with it, this is also something that's called kriging that has been around forever in kind of spatial stats. But folks don't tend to use it as much for like forecasting in time. This is a little bit of an unusual thing to do. And we're going to allow this heteroscedastic extensions, which I will explain in a moment, where we're going to let seasons have different amounts of variance. So now there's lots of different ways that one can define this covariance. And we're doing a really simple thing where we're going to assume that that this covariance depends a lot on how far apart any particular points are in our predictor space. And we're going to then, like this, I think this is all really standard stuff. It's super straightforward. Okay, but the thing that's kind of fun is what are our predictors? And like they are literally dead simple. So like the first one is just a count from 1 to 52 about which week of the season you're in. The next thing is the starting level. So you want to make sure that when you start the season that like it kind of should, like this week should be like last week. So if you give it the amount of observed cases in the last week of the previous season, that way everything kind of works together. We're going to put in a sine wave to try to enforce that season should go up and then come back down. Kind of a very simple assumption. And then we're going to add the severity indicator. And this was the thing that we found that we really needed to try to understand the huge amount of variation from season to season. And so what we did is for every season that we've already observed, we classified it as being low, -1, medium, 0, or high. And for San Juan, these were our thresholds. And we just chose these. Like we asked folks, what do you think would be a lot of cases of dengue in San Juan? Like what might be something you'd be concerned about? And we classified them that way. So it turns out that even with taking the square root and including the severity, we still had differences in the amount of variance between these seasons of different severity. So we needed a way to account for this. In fact, some of it was really big, especially like in Iquitos. Some seasons were so much worse than others that you had with huge swings, a lot more than what you would expect from that, and that can be taken care of with just our square root transformation. So what we did is the following. So we introduced an indicator variable based off of the severity level into our nugget, right, which allows that noise to be different in these different parts of our space, in our space, because this is a predictor, right? So that severity level is a predictor. So what it allows us to do then is effectively kind of stretch out the variance in those different severity level years. And this is also, when you do it this way, it's also able to be inferred in closed form. And it turns out that it works really well. So here's an example. So this is one of our seasons. And what we've done is we've colored the data to indicate the severity here. So this is a low season. The first season was low. These two are kind of our typical season, our medium season. And this season is one that we haven't observed yet. So all of those black circles are the real data that we haven't actually observed. We haven't included this in our fitting yet. And so given that we ended here and the sine and the cosine, and then we set the initial severity level, we estimate it, but we always give it a setting. And we purposefully set that towards severe, because if you're planning for dengue outbreaks, it's much worse to be wrong and underguess than it is to be wrong and overguess. So even though we knew that that might bias kind of our predictions, we figured that from a public health perspective, we would prefer to be biased in that direction. And so that's why we actually see in this case that we have a pretty big hump here at the beginning before we've learned anything. And then as we gather data, so here's week 16. So you can see now 16 of those little dots are full. And so we start like decreasing our variance around that part. And we're saying, oh yeah, we're really sure we're going into a high year. So we think that really it's going to be big. And we overshoot because we think that probably we haven't reached the peak yet. And in previous years, the peak was later when it was high. But this year it was a little earlier. Once we've gone over that bump, the model says, oh yeah, I think we're coming down, except it turned out there was one season that looked similar that had a second hump. And the model says, oh, I think maybe it could be like that season. And then by the time we get there at the end, we're doing a really good job. And we can kind of see too that we get, so here are estimates of like when is the peak going to be and how high it is, like those are our bars. And then here's the total amount divided by 52, so it would fit on the plot. And you can see as we go along, we get better and better estimates of those things. So everything works pretty well. And this was a competition, so we weren't the only ones trying to estimate it. So we can actually look at our model performance and everybody else's model performance. And here's a spaghetti plot to kind of give you an idea. So this is peak week and peak incidence for San Juan, where this is our log score, and our green one is our heteroscedastic Gaussian process. And being closer to zero is better. And so you can see some common patterns that pretty much everybody, not everybody, but most folks, as you gather more data, you get a better prediction, right? Which you better hope you do. Like if that's not the thing that's going on, that's a problem. But across weeks, like you go up at different rates, and not every model is equally good all the time. But if we look at aggregate across all weeks, it turns out that our HGP does really good. Well, so this is across all of. But this is for San Juan. They have all of our peak week of the peak and the season total across 3 different metrics. And the thing that I want to point out, which we were shocked about, is that the RHGP ours gets in the top 3 models on every metric overall. And we found this shocking, and as somebody who works with a lot of biologists, the biologists were really kind of upset about this. Like this doesn't include any information about any of the environmental predictors. It doesn't include any biology at all, like nothing at all. But sometimes this is going to be what you want, right? Because what we're doing here, the thing that we're observing is actually not biologically based. Whether or not people show up to the hospital has a lot to do about people more than the biology. Like you have to have the disease spreading, but you know, people's behavior, they're more likely to go to the hospital if like their friends are sick, if they know that there's an outbreak. There's a lot of things that influence whether or not people show up at the hospital. So to summarize, like this worked really well and surprisingly well. And some of the things that were really good about it is that it's fast, it's really data light. I only need that incidence data. I don't need any of those predictors, which is good because then I don't have to forecast weather into the future, which it turns out is hard. Past a couple of days. I don't know about here, but in Blacksburg, you really cannot trust the weather forecast more than like 48 hours. It learns really quickly, and it doesn't care what those underlying processes are. You can't get them wrong because there aren't any processes. But some cons of that, it's really context dependent. I cannot use the model that I fitted on San Juan for Iquitos. I had to fit a separate model for that, and I can't generalize to any other place. I can't say how things are going to change if I implement a control strategy. I can't tell you like what's going to happen if I change things. And I can't extrapolate. I can't say, is dengue cases in San Juan going to increase in 20 years, in the next 20 years on average? I can't say anything about that from this type of model. So can we be able to answer some of these questions if we go more towards the other side of the spectrum, that is thinking about this mechanistic approach? So again, coming back to our idea of malaria here, I said that if we kind of look at everything that's going on outside in this mosquito, we know that these are going to be sensitive to environmental conditions, but I didn't really say kind of how this happens. On the biology, what we tend to think about this as being is that it depends on what are called traits. It depends on the traits of a vector. So what is a trait? So I'm going to tell you what I think a trait is eventually, but I want you to realize that if you go and talk to a biologist, they might disagree with the definition I'm about to give you, because there is not a consistent definition in biology about what is a trait and how they're measured and which things count. Some folks are very adamant that some of the things I say are traits are not traits. They are demographic parameters, for example. There's a whole bunch of people who will disagree, but I'm going to give you my definition. And this is the one that we're going to work with. So I'm going to say that a trait is anything I can measure about an individual organism. It doesn't have to be reported about the individual organism. It can be aggregated over that population, but it's something that you could measure. So it could be physical. It could be what we call a performance trait, how fast you can fly if you're a mosquito. Or it could be a preference, like a behavioral preference, like whether or not you like to bite humans or dogs. So why might traits be important for understanding transmission? So if we think about kind of what is the thing that we want to understand about transmission, I'd say the thing we want to know for vector-borne diseases is how many infectious bites you get on hosts, right? So how many infected mosquitoes are able to go and bite people. And if we kind of build this up from a little mosquito and the things that we can measure about them, like our individual traits, there's also ones about how they interact with other organisms, whether or not they can find the person to bite, whether or not they can find flowers for nectar, because many mosquitoes also need sugar. So these together, oops, I went the wrong direction. These together determine what's called the life history of these organisms. And this life history then determines how many organisms you see in a particular place. Like, what is the vector density? Are you going to have lots of them today or not very many of them today? So then that vector density goes together with some other types of traits, things that we can measure, like encounter rate. So how that comes, this one comes from a trait, whether or not this mosquito is going to manage to find you. And then if it gives you a bite, whether or not it can transmit it. So then all that together, we only get our infectious bites. And it turns out that we can represent this type of thing mathematically. And in the epidemiological literature, we write down an equation, and we usually call this equation R naught. It used to be that no one knew what R naught was. In the last 3 years, for some reason, now everybody knows what R naught is. So I don't have to explain using Kate Winslet and Contagion anymore, because now folks have heard of R naught. And so this is just the expected number of cases that you get when you introduce a new infected person into your population. And this is, again, like I said, malaria is the canonical vector-borne disease. And this is the equation for that, for R naught, for that kind of canonical setup. And what I want you to notice, I mean, it's not really super important what are the particular things that go into it, but there's a bunch of these things that are traits or that come from traits. So like the mosquito population comes from traits. These are all traits of the mosquitoes, and they all depend on temperature. Some aspects like the human population size and the human recovery rate don't, but other things do. And luckily, we actually know a lot, it turns out, about how biological processes vary with temperature. So usually what happens is the following. So we get something that's like this. So we have some sort of trait, and we measure the performance of that trait, and that's going to be our Y, and then we have temperature on the next. And we usually kind of get this hump-shaped curve. So for example, let's imagine that we're looking at eggs that are being laid. So if it's too cold or too hot, all those eggs die. No mosquitoes, no insects, or whatever, right? But then at some intermediate temperature, often this performance trait is going to be maximized. You get lots and lots of eggs if the temperature is good. And so what we want to do is we're going to go ahead and try to actually build a mathematical model that includes this and link it back into that transmission that we had on the last page. So here's what we do. Here's our approach. Our approach is to go and find as much data on these physiological processes that folks have taken using really time-consuming experiments in the lab. We're going to fit nice Bayesian fits to these, so we get some quantification of the uncertainty. And then using a technical term, we're going to mush them together using the R naught equation as really combine them. So each of these becomes a function of temperature so that we get R naught as a function of temperature. And then we're going to see whether or not this matches up with field transmission, whether or not the things that our predictions about when and where which temperatures are good for transmission actually holds up with what we see in the field. So we're going to look across two species of mosquitoes that are both Aedes species mosquitoes, Aedes albopictus, the Asian tiger mosquito, which is kind of a temperate mosquito. It doesn't like it to be very hot. And then we have this tropical species called Aedes aegypti. And these are two of the most dangerous animals in the world. They're responsible for the death of millions of people every year. They're only like second and third to things like Anopheles gambia, which spreads malaria in Africa. So these are really, they spread a lot of disease. So it's really important to know as much as we can about them. So folks have done a lot of empirical studies on these, but they don't always vary temperature. So actually we had, and this is the other reason to use a Bayesian approach, is that this is a lot of data. So do you see these dots here? These are well covered curves. Like this is the best data that we get for these systems. And we're looking at like 10 data points for any particular trait. For some malaria vectors, you get 4, and you're trying to fit these curves. So here are the traits. So these are some of our examples. This is just for Aedes aegypti. We have a similar set of curves for Aedes albopictus. And we have some things like, this is how much they bite. We have how many eggs they lay, how long they live, how likely they are to transmit it if they're infected, how likely they are to pick it up if they take a blood meal that has the infection in it, and then how long it takes for them to become infectious. So then we can now take all of these together and put them together with that R0 equation, and we get 2 different curves, one for each species. So here's the 2 curves that we get. So here, this light blue one, this is our temperate mosquito, and this dark blue one is our tropical mosquito. So first, great, our tropical mosquito likes it hotter than the temperate one. So we have a really good, like, just making sure that we're not doing something really wrong. So that's a good kind of first step. But we can see that we actually make different predictions, right? So that they both kind of we would expect not to have very much transmission if it gets too much below like 20 degrees centigrade. But we think that the albopictus really won't be able to transmit above kind of 32, whereas it's closer to 35 for aegypti. And that's a pretty big difference like that's 3 degrees centigrade, right? That's a lot. You notice those temperature differences as a person. So does this match up with what we see in the field? So what we did is we took some data. These are Aedes species. So we looked at data on Zika and dengue. We looked at the temperatures that were going on when you start having transmission going on in the field. And we plotted those along with these curves. And we find this. So this is areas where dengue and Zika and chikungunya, actually in here too, are actively being transmitted. And we can see that, of course, it's not going to be perfect because actually classifying whether or not transmission is going on is tricky. People bring in infections from other places, for example. But you can see pretty much that like we're not getting transmission in the places where we think the transmission should be low. And we're getting a lot of transmission at the temperatures where we think transmission should be high. So that's a really good proof of concept. So can we do anything else to kind of validate this? So we've tried a few other things. It turns out like validation, like more than this kind of qualitative validation, is a little bit tricky because what does it mean for your model to be valid? So we've tried a few things, like we use R0 as a predictor in a GLM to say, can we predict yes or no whether or not there's transmission going on in a location? And then can we use it to predict the incidence scaled by the population? Because again, it doesn't matter what R0 is, what the temperature is, if there aren't any people there to get disease. So if you have a higher population and a higher R0, you would hope that you would predict that there's going to be more cases. And actually we predict pretty well. Our classification rate is high and our mean absolute percentage error is pretty low. So let's say that we trust that what we're doing is reasonable. Now what can we do with this model? So what we decided to do and give this a try is, again, we're interested in kind of both when and where we're seeing transmission. So we said, okay, if we look at those those blue curves, we fit everything with the Bayesian approach, so we can calculate the posterior probability that R naught is greater than 0 at every temperature. And we want to be really sure that R naught is greater than 0. We want to be pretty sure that we're not completely excluding transmission at that temperature. So we set a really high threshold of like 97.5%. We're calculating the probability that the posterior of R naught is greater than 97.5%. And we get this region, right? And we're going to call this the permissive transmission zone. I also call this the Goldilocks zone for our transmission. And we're going to calculate at every location over the whole Earth how many, or although, and also in the Americas, I'll show you the Americas first, and then we'll show you some global stuff afterwards. How many months of the year on average are you in this kind of sweet spot? So here's how it is kind of a historical one. This is from a few years ago. So this was the initial case, again, for these two different mosquitoes. So we have our temperate one over here on the left and the tropical one on the right. And the deeper red here is 12 months to 0 months. Effectively, actually, it's white, but effectively every place you see gray is 0 months. And we can see that the tropical ones, like Brazil, is like great big hotspot for the tropical mosquito. But actually, there are some spots of Brazil that are too hot for the temperate mosquito to be able to transmit. So it's in some cases, it's about the mosquito not being able to survive. In other cases, it's about pathogen not being able to be transmitted. And if we look at where the CDC says these mosquitoes are, which are both invasive mosquitoes are, it corresponds to about kind of 4 to 5 months of potential transmission. And then it keeps going up, right? So we have a little bit up here. And I don't know how many of you have noticed this, but we had local transmission in New Jersey this year, which this tells us that it's probably just about that it happened to have gotten here. Then it's not that the climate has changed substantially. There used to be malaria in New York, right? So it's about that we happened to have gotten it and we got kind of unlucky. And then we can look at how we expect these ranges to shift under different climate change scenarios. So for example, specifically looking at dengue here, this is kind of the current temperatures versus the projected temperatures in 2080 under 2015, 2080 under a business as usual scenario. This is the most climate warming. And what you can see is that you have a lot of expansion predicted in areas that have suitable temperatures towards both poles, but then like in the middle, potentially, even for these tropical mosquitoes, it could end up getting too hot in certain areas by 2080. But you're going to make up for it in some expansion. So that's in space. What about for people? Like, what does this mean for the people who might get sick? So if we calculate the number of people, of new people that are going to be living in places where the temperature is going to be good for transmission around the world, across multiple scenarios, we're going to get something like this. So let me take you through one of these boxes very quickly. So here we have our tropical mosquitoes here. We have 4 climate scenarios from the least warming to the most warming. And then what we have here is the 2050 and 2080. And this is the change in the number of people who live in places that where the temperature is going to be changing. And the axis is in billions of people. So we're saying that under the top climate change scenarios, 1 billion more people will be living in places where the temperature would be ideal for at least 1 month of transmission, right? That's a huge expansion in the range and the number of people that could potentially be affected. It's not as bad if you look over a whole year. This one's a whole year, but we're still looking at like increases in the number of people affected on the order of 250 million people living where the temperatures are good for transmission, in that case year round. By Aegypti, which is our tropical mosquito. For our temperate mosquito, we still have a big increase in places where there's any transmission, but it starts going like over the hill because it's getting way too hot in places that historically had both a lot of transmission and a lot of people. And so actually we have a net decrease. could say, is that a good thing or not? I don't know. And it turns out, though, too, that we can also make different predictions for different diseases. So this one is using only the traits that are specifically for dengue. So this includes the mosquito traits plus the transmission traits for dengue. We can make different, I can expand, I can plug in a new trait for Zika, and I can make a new prediction for Zika about how transmission might change for Zika, which is being transmitted by Aedes aegypti. So it becomes kind of really nice and extensible. I can make lots of predictions. I can say, okay, how many people in different areas am I going to add who've never been exposed to Zika? How many people might be exposed to Zika in different regions and different times under different climate change scenarios? So for this one, I can do a lot of kind of predicting into the future. I'm doing a lot of extrapolation. I have, because I'm basing this on a strong belief about the underlying biology, right? I'm not having to, and what folks often do is they instead fit correlative models and say, where do I see dengue now? And what are the environmental conditions now? And then extrapolate, which we all know can be problematic as those predictors shift and change under the hood. So this one we're saying, as long as we believe that climate change model, we can put some limits on kind of where we think the temperatures, everything else being equal, what temperatures are going to be allowing or prohibiting transmission in different areas. And how many people might that affect? So I've kind of gone for two extremes, right? So I went, the first thing I said, no biology at all tactical. And this one isn't really fully strategic. If I showed you an SIR model and just like put some random traits in there and like did some forcing and showed you what those patterns look like, that would really be on the fully strategic end of things. So this is trying to move a little bit more into seeing how we can bring these things together. And I think there's a lot of opportunities. In my lab, we've been trying to take kind of this bridging approach by doing things like taking these traits and metrics and using them as predictors in things like GLMs in order to do forecasting, or for what we call an ecological niche model, which if any of you grad students ever read that, it is literally just any correlative model that you build to try to predict whether or not something's there. So it could be a GLM, it could be CART, it can literally be anything. That's just what the ecologists call any model that does that. We're also trying to think about how we can use this trait information as priors to constrain the shapes of responses or the limits of these responses for either focal or related species, because it turns out some species are really easy to study and some species are hard to study in the lab. There's this one called a tsetse fly, which if you have a strong stomach, I encourage you to Google what a pregnant tsetse fly looks like, and it's pregnant. They're like little kangaroos. They have like a little pouch, but they are wild looking. But they spread African sleeping sickness, and it turns out they are really hard to raise in the lab. So our data on those are poor. But we have data on other flies that are related. And so one of the things we want to be able to try to figure out how to do is how to use that data on the things that we can measure well, but the things that we can't measure well, so that we can, again, start trying to kind of bridge these gaps and put constraints on things. So I think that these different types of approaches, the tactical and strategic, give really nice complementary predictions. So for the first one, I couldn't tell you what was going to happen in a different location. I couldn't tell you like what would happen in space. The second one, I can tell you what's going to happen on space, but I couldn't tell you how many people are going to get dengue based off of that model. I just can tell you that I think it probably would be more or probably be less if you don't do anything to try to control it. So they're really complementary predictions, for example. And I think that if we start figuring out even better ways to start combining them, that we'll be able to do things like say, well, what might happen in 5 or 10 or 15 years, instead of looking at 2050 and 2080, which is what I did for all of those maps, and start to actually be able to kind of get predictions at timescales that are useful for people like policymakers about where should we spend our money to try to stop the spread of dengue, for example. And I think that there's a lot of opportunities here for statisticians, because you've seen that like the models that we're actually fitting are not complicated models. It's using a lot of stuff that we feel and we get used to as being standard tools that are not standard in these disciplines, but they're really useful. But the thing we do need, and I'll be preaching to the choir here, is that we need the right types of data. We need good data, right? So it turns out to do these sorts of things, we need data on the traits. We need data on the vector dynamics. So you need to be able to have folks going out and collecting data on how many mosquitoes you have. You need case data. And right now in this field, unlike kind of, for example, machine learning, where you have kind of standard data sets that are open that everybody uses to compare their models and methods, we don't have that here, and it would be really, really useful because then we'd be able to actually tell which models are doing a better job. Right now, it's almost impossible to compare them. And so one of the things that I try to convince folks of is these things, right? So that when you're building your data, and so if you're ever working with someone who is generating data, this is what we're trying to convince them to do because it will make their lives easier long-term and our lives. You want it in summarized and standardized formats, because right now, if you go and you try to do this, you'll notice it's not, and it makes it hard. And everything really needs to be a lot more open so that we can actually make progress. And this will allow us to actually be able to assess how well we're doing. And so we've convinced the NSF to put some money into helping us do this. So we've been building databases to hold the type of data that gets used here for vector-borne diseases. So we have a new database on traits called Vet Traits. It is now out of beta and it is ready for accepting data, which also means that there are data there that y'all can play with and y'all can start building stuff on. We're also doing, we have our VecDyn, which that one's still in beta that we're working on. We are trying to start building online tools and some open source code to allow you to interact with this and fit all these different models. And we've been building tutorial materials on mathematical and statistical methods. So primarily aimed at biologists, but at least it's all open. So you guys can take a look if you're keen, and that's at vectorbyte.org. So that's all that I have to say. Thanks very much for your time.
Do you have any questions here?
So when you are picking the model, did you use the observed data to calculate export, the severity measure?
So yes, so the log score uses both our predictions and the distribution of predictions and the true data. So in that case, we had the data, we just weren't allowed to use it to fit the model. In fact, what they did is they gave us the first, like up until a certain date of all the data, and then they held back the data for all the others. And then you were given, they like handed you the rest of the data and you were given like another 48 hours or something to regenerate your predictions on the new data and train as you go. So it's possible that somebody cheated, but we didn't cheat. We only used the data that was allowed. But yeah, that's how, like all the log score always has to have the truth there, or you can't score it.
Another thing is I start online. Those mosquitoes you are talking about, those are tropical mosquitoes. One is tropical, another is
it's more temperate. It doesn't like it quite as hot. Yeah.
then in Alaska, a lot of mosquitoes, but they do not spread the disease.
Yeah, so actually I'm always kind of amazed that vector-borne diseases exist. They're almost always like, there's only a couple of mosquitoes that typically can spread any particular one of them, and they often can only be spread to specific types of hosts. So like there's a bunch of different types of malaria, but there's some kinds that only go for mammals and some types that only happen in birds, right? And then you have these Aedes species that spread a lot of these different viruses. So we've seen, and there's a lot, I mean, there's so many of these different types of mosquitoes. But so for example, West Nile virus is the same, it's a very similar virus to dengue, it's in the same family. It's also spread by these Aedes mosquitoes. But in that case, like, we are really bad at reproducing enough West Nile, so like we are dead end hosts. Like, you get it, you don't pass it on. Actually, it turns out it doesn't matter how many mosquitoes bite you. After you get it, you're not going to pass it on. It's the birds that pass it on. We just happen to get it because it's unlucky. Whereas for malaria, that malaria will not get passed on if it is not in a human host. So if that mosquito doesn't bite mostly humans, and some of them will only bite people, some of them are super finicky and will only bite living people. Like, you can't give them human blood and not in a person. So be really glad that you're not a biology grad student who is needing to feed mosquitoes, because I know some of them that do that, and I wouldn't want to do it, but it is a thing that happens. Or are there biology grad students here? I'm sorry. Hopefully you guys don't have to feed your mosquitoes. They've gotten a lot better at figuring out, like, they now have like these membranes that the mosquitoes are happier to use, for example, but there are still some colonies that are fed by humans. But that doesn't require special permission, because apparently, like, you don't have to have human, like when we do surveys of statisticians and you have to get permission to interview your humans, it turns out that grad students that are volunteering, they don't have to like have the same permission as asking questions. I find that wild, but anyway.
Oh, yes, I did have a question. So whenever you were mentioning kind of like the different traits that you were putting into the models, so things like biting frequency, is there any way for you guys to account for like if there would be any level of like vector manipulation by the pathogen? So like, are they more likely to bite if they're infected? Or...
That is a really great question. And for those of you who are not familiar with this, many pathogens do modify the behavior of individuals that are infected with them. And there's not enough data for most mosquitoes to be able to determine if that is happening. We think it might be happening in certain cases. That at least it seems that their preference changes. We know it happens for aphids. Aphids also spread diseases. They spread diseases to plants. Aphid behavior gets changed when they're infected. They're much more likely to bite they like sick plants when the aphid is healthy, and they like healthy plants when the aphid is sick. So that actually increases the rate of transmission for those types of things. So you can actually put that stuff into the model if you have data on it. But the tricky part is actually getting the data. And biting rate of mosquitoes is really, really hard to measure. So actually being able to tell what's going on is hard. So for some species, they make the really ridiculous assumption that the amount of time it takes for a female to produce eggs is the interval between bites. And so you just go 1 over that, which from a data perspective is not great. But it turns out that actually measuring anything else is really hard. Yeah.
So from an etymologist perspective, I'm always thinking control. Like you mentioned, the model can predict if nothing has been done for a couple of years, what will happen. But in reality, I would assume there are always something about control of inflation to put out mosquitoes, ticks, other vectors. So how do you think the control aspect can be added to the model?
So either you would have, so if you wanted to be on the strategic end of things and you wanted to include control in your model, you would have to actually like have different places or different times where you've implemented the controls and then like fit your strategic model across those to try to get it at what the dynamics might be under these different sections. On the more, that's the tactical end. On the more strategic end, if you're building this mechanistic model, then you would say, okay, I know that when I spray, for example, for mosquitoes, I'm going to increase the mortality rate of adult mosquitoes. And that's one of my parameters in the model, and I can decrease it. And I could choose, if I wanted to spray at particular temperatures mostly, and affect only parts of that curve. And you might actually decide that that's the best thing to do if you have limited opportunities to spray. Right? You don't want to spray all the time. You don't want to spray when there's not any mosquitoes. So you would necessarily do it at particular times of year, and you can go ahead and put that into the model. And folks do that. And actually, because this is the other reason not to rely purely on a correlative approach, because your controls are correlated with your environmental conditions and you're artificially decreasing incidence in places where the environment is good for transmission, you can actually misestimate the relationship between your input and your output when you actually have that going on. And so then you're from a statistical perspective, like all of your estimates are not so good, which means that even making your regular predictions are tough, let alone trying to either forecast or extrapolate out. So I really like the fact that like being able to bring this stuff in and trying to use it to help improve our ability to do that kind of to figure out what the relationships are.
So do you incorporate spatial aspects in your model, like vector movement, movement?
So we haven't been yet, partly because of the scale of the data that's available. So often what we have information about is stuff that's at like kind of 5 kilometer scales. And most mosquitoes don't travel quite that far. They can travel a lot, and if it's windy enough, they can travel really far. But we don't usually go ahead and incorporate that. So instead, we're really looking at larger spatial scales. So if we wanted to do smaller spatial scales, which is something we would like to do, like villages, like we're working with some folks on again aphids for like potatoes and maize in Africa and South America, and they're very interested in kind of being able to build better local predictions. And in that case, you really need to know where those aphids are going to move, because they will move and they're going to take their diseases with them. So you really do want to do that. But right now, that's the type of data that we have access to doesn't allow us to do that.